Think about someone you love, a parent, a grandparent, someone whose face you know better than your own. Now think about what happens to that face over time. The lines that form, the slowness that sets in, the day they forget something they never would have forgotten before. We call it aging. We call it natural. We call it inevitable. David Sinclair calls it something else. He calls it a problem, one that can be solved. And in January 2026, for the very first time in history, that solution was injected into a living human being. This is what happened and why it changes everything. To understand what Sinclair discovered, you need to understand one thing about your DNA. Your DNA is not the problem. Every cell in your body contains the same DNA, the same 3 billion letters of genetic code you were born with. That code doesn't change, it doesn't decay, it doesn't age. What changes is something else entirely, something called the epigenome. Think of your DNA as a compact disc. The information on it is perfect, every song, every note, exactly as recorded. But over time, the disc gets scratched. The reader can't interpret the information correctly anymore. The music skips, the data gets lost. The epigenome is that reader. It's a system of chemical signals that sits on top of your DNA and tells each cell which genes to activate and which to ignore. A liver cell and a skin cell have identical DNA, but completely different epigenomes. That's what makes them different. And as we age, those signals deteriorate. Cells start misreading their own instructions. A liver cell forgets it's a liver cell. A neuron forgets it's a neuron. The information is still there, but the reader is too damaged to use it. Sinclair calls this the information theory of aging. Aging is not wear and tear. It is not inevitable decay. It is a loss of information. And if information can be lost, it can potentially be restored. For most of human history, the idea of reversing aging was mythology, the fountain of youth, the philosopher's stone, immortality as the obsession of kings, alchemists, and gods. 200 years ago, the average human lived 35 years. Not because aging was reversed, but because we got better at surviving. Vaccines, antibiotics, surgery. Modern medicine doubled human lifespan by defeating the diseases that killed us early. But aging itself was never touched. Medicine learned to treat the symptoms: cancer, heart disease, neurodegeneration, while the underlying clock kept ticking. The scientific community, for most of the 20th century, treated aging as a fact of life, not a disease, not a problem, simply biology doing what biology does. David Sinclair disagreed. In the 1990s, working at MIT under Leonard Guarenti, Sinclair began studying why yeast cells age. He found that the same genes responsible for aging in yeast, the sirtuins, also existed in humans. The biology of aging was conserved across species. That meant it was not random, it was programmed. And if it was programmed, it could be reprogrammed. Decades of research followed. Animal models, molecular mechanisms, paper after paper. And then, in 2020, the experiment that changed everything. In 2020, Sinclair's lab at Harvard published a study in Nature that the scientific community had never seen before. Using 3 modified genes, OCT4, SOX2, and KLF4, known together as OSK, researchers injected the therapy directly into the eyes of old, blind mice, mice with glaucoma, mice whose optic nerves had been destroyed by age and injury. Within weeks, they could see again. Not partially, not temporarily. The cells of their retinas had been reset, the epigenetic damage reversed, by up to 75%. The cells had remembered who they were. In 2023, the team went further. Publishing in Cell, they showed that by controlling OSK expression, they could speed up aging in mice and then reverse it. Aging, for the first time, was not a one-way street. The question then became, does this work in humans? In January 2026, the FDA answered that question by granting permission for the world's first human trial of epigenetic reprogramming. Life Biosciences, co-founded by Sinclair, began phase 1 studies of their therapy ER-100 in patients with glaucoma and NAION, a rare condition that destroys vision by damaging the optic nerve. The trial is designed to test safety first. Results are not yet available, but for the first time in history, a human being received an injection designed not to treat the symptoms of aging, but to reverse its cause. Sinclair watched his mother develop lung cancer at 50. 20 years of diminished life followed. He has spent his career asking one question: what if that didn't have to happen? We don't have the answer yet. The human trial is in its earliest stages. Longer-term data doesn't exist. The full scope of epigenetic reprogramming in humans remains unknown. Science is honest about that. But here is what we know. 200 years ago, we lived 35 years on average. We didn't accept it. We fought back. And we won. Now we are asking a harder question. Not just how do we survive longer, but what if aging itself is optional? A scratched disc can be polished. A cell that has forgotten who it is can remember. For the first time in history, that is not a metaphor. It is a clinical trial. Stay curious.